Should I start sharing the screen or wait a bit more?
Yes, you can start sharing.
Okay.
While we wait a few seconds. Okay, going to full screen, covering my notes. But anyway, so while people is joining, I just would like to introduce today's talk more generally. So first of all, welcome the Imaging One World lecture series organized by the Friends of the Royal Microscopical Society, together with Stephanie, Alex, Nick, and Curti, to keep the community interacting despite the ongoing pandemic. It is a great honor to welcome to Imaging One World Professor Jin Zhang, who agreed to present despite being 5 a.m. in California. Jin received her PhD in chemistry from the University of Chicago, and after completing her postdoctoral work at the University of California, San Diego, she joined the faculty of Johns Hopkins University School of Medicine in 2003. She was then promoted to professor in 2013, and in 2015, she moved back to UCSD, where Jin is a professor of pharmacology, bioengineering, chemistry, and biochemistry. So Jin Zhang's laboratory is internationally well known for the enabling technology they develop to probe cell biochemistry in living samples and their applications to the understanding of the molecular mechanisms underpinning cell physiology and diseases, including cancer. So then, thank you very much, Jin, for joining us today, and feel free to start whenever you're ready.
Great. Thank you very much. Thank you, Alexandra, for the introduction and also for the invitation to speak. I'm excited to share some of our recent work with you all. So my lab for the last 18 years has been interested in understanding how the cell organizes activities to achieve very specific functions. At the molecular level, biochemical activities such as shown here, represented by protein phosphatase and kinase activities, are underlying and these biochemical activities are also interconnected and integrated. In a complex network like this, each of these connections is made through protein-protein interactions, small molecule protein interactions, and enzymatic reactions. And this type of a complex networks are also embedded in the spatial temporal framework of a living cell, being constantly modulated. And my lab has been interested in understanding how these activities are organized inside the cells. And we, from our recent work and work from others, we raised a new hypothesis. We hypothesized that cellular biochemical activities are not floating inside the cells. Instead, they are spatially organized into an activity architecture. And we further hypothesized that disease states, such as cancer, reflect the reorganization and restructuring of this activity architecture. And we'll be interested in developing new microscopy methods and also new fluorescent biosensors to go into the living systems and probe this biochemical activity architectures across different scales, from the molecular, nano, micro scales, all the way to subcellular and whole organism level. So shown here is one type of fluorescent biosensor technologies that we are developing in the lab. We're developing kinase activity reporters. We call these CARs. And as you can see, we're basically generating chimeric proteins that serve as kinase activity reporters that allow us to track kinase activity in living systems. And these chimeric proteins are made of quite a few different components. Two of these components are a pair of fluorescent proteins shown here as cyan and yellow fluorescent proteins. They, of course, have the appropriate spectral overlap so that they can serve as a FRET pair or a pair of fluorescent protein that can undergo Foster resonance energy transfer. And of course, the efficiency of a FRET is dependent on the distance and the relative orientation between the two fluorophores. So together, they serve as the reporting unit of our biosensor. And we spend a lot of time engineering the middle portion, which we'll call the sensing unit. And for kinase activity reporters, the sensing unit consists of a substrate peptide and a phosphoamino acid binding protein or binding domain, which is a module that recognizes the phosphorylated form of the substrate peptide. So the idea is when the kinase is active, it comes in and phosphorylates the substrate peptide. And then the phosphorylated form of the substrate peptide will fold into the binding pocket of this binding domain, thereby synthesizing a conformational change, bringing the two chromophores close to each other. And we now get a change in Fred. So during the experiments, we excite the donor fluorophore and look at the emission from the donor and emission from the acceptor, and then calculate the ratio as the readout for Fred. And this allows us to track the kinase activity in living systems. As you can see, we have used this design to generate a lot of cars. We started with protein kinase A and have generated reporters for PKB, PKC, PKD, ERK, and more recently for mTOR complex 1. We have also used this reporter design to generate the sensors for phosphatases. And this design can also be applicable to generating sensors for second messengers. Shown here is our indicator for cyclic EMP using EPAC. We call this IQ. It's a sensor for the ubiquitous second messenger cyclic EMP. For generating IQ, we simply took a cyclic EMP binding domain or binding protein that changes the conformation upon binding to cyclic EMP, and sandwich that between a Fred pair, shown here as a cyan yellow pair. And the cyclic EMP binding induced conformational change can be translated into a change in Fred, which allow us to quantify the free intracellular cyclic EMP levels and also monitor cyclic EMP dynamics inside the cells. And we have also used this type of designs to generate sensors for lipid second messengers. Shown here is our indicators for phosphoinositides. We have used the plaque homology domain as the part of the sensing domain, sensing unit for sensing these lipid second messengers. For example, we use the plaque homology domain from AKT to bind to PIP3 and the PI3,4 bisphosphate. These are 3 phosphoinositides that regulate a lot of different cellular processes, such as cell growth, proliferation, and migration. However, when we try to use the plaque homology domain to sense phosphoinositide, we encountered a problem. As you can see, this is the pH domain without the ligand and with the ligand. It doesn't change its conformation much upon binding to the ligand. So what we did is to engineer a pseudo ligand, basically a stretch of acidic residues, to pre-associate with the binding domain or binding pocket of the binding domain. And when the real ligand come in, it will kick off this pseudo ligand, and that's our engineered conformational change to be translated into a change in Fred. So this design allows us to monitor these phosphoinositides, these 3 phosphoinositides, and also more recently PI4,5 bisphosphate. For example, we also generated a color variance for these 3 phosphoinositides and PI4,5 bisphosphates, which allow us to look at these 2 types of the lipid second messengers simultaneously inside the cells. I won't have time to go through some of the other biosensors that we developed in the lab. In fact, in the process of preparing a review article a couple of years ago, Eric Greenwald, a former postdoc in the lab, put together a biosensor database, and this is the website. And this basically is a long list that includes a lot of different biosensors generated by us, but also by a lot of people in the field. So turns out there are more than 800 variants of different biosensors that are already available. So I encourage you to check out the list and let us know if we missed some of the biosensors that are recently published. So these fluorescent biosensors serve as powerful tools for us to probe spatial temporal regulation of cell signaling in living systems. Shown here are a few different examples where we look at a spatial control of cell signaling at different organelle level. We've looked at a new pool of mTOR complex 1 in the nucleus and how this nuclear pool of mTOR complex 1 is regulated. And this is work done by Jin Zhang, a project scientist in the lab. And Mingyuan or Shang recently also looked at a lysosome selective regulation of these 3-phosphonosides and AKT in different living systems. And this work is in press. Shang is a current graduate student in the lab. A former postdoc, Jeremiah, also looked at a non-canonical regulation of ERK by RAP1 at the plasma membrane and discovered a new functional role of these plasma membrane ERK. And this work was published last year. But today, I wanted to focus on one specific example to showcase how these biosensors can be useful tools for us to understand the spatial temporal regulation of cell signaling. And this particular example is focused on cyclic PK signaling. In a classical model, GPCR activation leads to activation of a trimeric G protein. For example, some of the GS-coupled receptor will lead to activation of a G-alpha S, which dissociate from beta gamma and activate the adenylate cyclase, the enzyme that produce cyclic AMP. Cyclic AMP then binds to the regulatory subunit of the tetrameric holoenzyme pKa, thereby unleashing the catalytic subunit. And the CAT is the active unit, then which then phosphorylate a lot of different substrate to regulate a variety of different cellular processes. Because cyclic AMP is this ubiquitous second messenger that connects a lot of different GPCRs to a variety of different cellular processes, the cell really has to have a mechanism in place to make sure this cyclic AMP signaling is highly specific and very locally regulated. And in fact, close to 40 years ago, a model of cyclic AMP compartmentation was proposed to help explain the highly specific cyclic AMP signaling. In this particular model, it suggests although cyclic AMP is an intrinsically freely diffusing small molecule, its signaling effect is highly local. And in this model, this enzyme phosphodiesterases, these enzymes that degrade cyclic AMP have been suggested to play a critical role of locally degrading cyclic AMP and restricting cyclic AMP diffusion. However, there's a big problem with this model because these PDE molecules or PDE enzymes are not very active. If you look at the in vitro numbers, they do a few turnovers per second, so they're relatively slow enzymes. And for these enzymes to to restrict the cyclic AMP diffusion and create cyclic AMP nanodomains or microdomains inside the cells, cyclic AMP diffusion within the cells really have to be slowed down by 2 to 4 orders of magnitude compared to these parameters identified in the literature. So these PDE molecules are definitely not as effective as this shark in terms of creating these nanodomains or microdomains. So we recently discovered a new mechanism for these cyclic AMP to be highly compartmentalized inside the cells. And turns out that one of the regulatory subunit of protein kinase A, R1 alpha, phase separates and serve as dynamic buffering systems to help create these cyclic-MP compartments. And this is work done by Jin Zhang, a former graduate student. Our work started with our interest in these functionally non-redundant regulatory subunits of a pKa. There are 4 different isoforms shown here, and we're particularly interested in R1 alpha, which is ubiquitously expressed in all tissues. And this is the one, when we knock it out, the animals exhibit embryonic lethality. To start with, Jason was just interested in looking at R1 alpha at the endogenous level. So he took an approach developed by Bo Huang's lab at UCSF, used the superfolder GFP, separated into 2 fragments, FP11 and FP1-10. This smaller fragment, which corresponds to the 11 beta strand of the beta bara, which they call FP11, is knocked onto the endogenous locus of R1 alpha. So this makes a fusion of FP11 to R1 alpha. Then Jason co-expressed the FP1-10, and these 2 fragments will just find each other and reconstitute into a functional fluorophore that allow us to look at R1 alpha at the endogenous level. So when Jason did this experiment, he saw 2 distinct pools of R1 alpha molecules. Some are diffuse and some form these puncta. It turns out these puncta can move around a little bit and merge occasionally, as you can see here, and also in this short movie, the 2 puncta merge together. Using fluorescence recovered after photobleaching, where we were able to show these R1 alpha molecules can freely exchange between the diffusible pool and the puncta. So these characteristics reminded us of liquid-liquid phase separation, which attracted a lot of recent attention from medical research field and biophysics and biochemistry field. So we know a lot of the proteins, if they have intrinsically disordered regions, they tend to phase separate. We also know phase separation is quite often driven by multivalency. We do note in a lot of cases, we observe these proteins or these molecules exhibit this behavior, but we don't know what kind of biochemical activities and functions that will emerge from these phase separation process. Since we suspect this is liquid-liquid phase separation, we did what everybody does in the field, treated these cells with 1,6-hexenediol. And this indeed disrupted these R1 alpha phase separated bodies, shown here by the image and the quantification here. Also, using purified R1 alpha molecules that we obtained from Susan Taylor's lab, we were able to show these purified R1 alpha molecules can form these liquid droplets. And depending on the solution conditions, they can form these liquid droplets at a concentration as low as a couple micromolar. Interestingly, presence of the catalytic subunit can inhibit this process. If you look at the y-axis, this is increasing concentration of pK-cat. So with increasing concentration of cat, we get more of the red circles showing up. This indicates no droplet formation. And even more interestingly, the presence of cyclic-MP can reverse this inhibitory effect of cat, or the catalytic subunit of pKa. So with cyclic-MP present, we got more of the blue circle showing up. We think we understand to some extent where this cyclic-MP responsiveness comes from. Because from the early truncational studies Jason did, we know this N-terminal AD domain, this dimerization docking domain, and this linker region are necessary. and to some extent sufficient for this phase separation process. From the structural studies done in Susan Taylor's lab, we know cyclin MP binding to these cyclin MP binding domains will make this intrinsically disordered linker region even more disordered. And this linker is also more exposed when the catalytic subunit is unleashed. We think this drives the phase separation process. And indeed, in cells using FP tagged R1 alpha and the PK CAD, we see cyclin MP dynamics dictate the R1 alpha phase separation dynamics. When we induce cyclin MP production by either treating the cells with phospholene or isoproterenol to stimulate cyclin MP production, we see more R1 alpha punctal formation, as you can see in this movie. Interestingly, although in this classical PK activation model, CAD is unleashed upon cyclin MP binding to the R1 alpha, we do see these catalytic subunit get recruited to the R1 alpha phase separated bodies. So they show very good co-localization. I can come back to this during the discussion if anyone's interested, but we think this gives some evidence for a non-canonical activation model for PKA. But now, for now, we show cyclin MP dynamics dictate the R1 alpha phase separation dynamics, and we're interested in now understanding what type of biochemical activities are present in these R1 alpha phase separated bodies. To do this, we developed a new class of fluorescent biosensors, which we call Fluorescent. These fluorescent biosensors targeted to endogenous proteins. So we basically built down this split FP technology to generate split biosensors. We took our FRET-based fluorescent biosensors and take this donor fluorophore, which is based on superfolder GFP, and separate that into two fragments again. And this beta, the 11th beta strand, the FP11, again is fused to protein of interest by using CRISPR-Cas9 technology. And the rest of the biosensor will be co-expressed. And the rest of the biosensor contains the FP1 to 10 and the sensing unit and the donor, excuse me, the acceptor. And now when these two fragments of the donor find each other and reconstitute into a functional donor, now we get our functional biosensor back. Because again, no donor, no readout for a FRET-based biosensor. So this allows us to monitor signaling activities next to a protein of interest at this endogenous expression level of the protein of interest. So we applied this technology to R1 alpha and also more recently applied it to a variety of different biosensors and to several different proteins of interest to demonstrate this is a general methodology that can be used to look at signaling activities at the endogenous level. Here, I'm skipping some of the data, flow step data for R1 alpha, just to show one slide here. We looked at a PK activity and also cyclin MP dynamics in using this flow step technology. Indeed, these newly formed R1 alpha phase separated bodies contain higher cyclin MP levels and also higher PK activities. In fact, even in the resting state, these phase separated bodies contain very high level of cyclin MP, and the cyclin MP sensors in these phase separated bodies are already saturated even in the resting state. So these data allow us to propose a new model where we suggest that R1 alpha phase separated bodies actually sequester and buffer cyclin MP, serve as a cyclin MP buffering system to enable cyclin MP compartmentation. So when cyclic MP goes up, there's more punctal formation, and cyclic MP get sequestered and buffered into the phase-separated bodies, thereby lowering the amount of cyclic MP outside these compartments. And now PDE molecules can do a much better job of locally degrading cyclic MP to create these cyclic MP nanodomains or microdomains. And this is critical for effective cyclic MP compartmentation. I won't have time to show you the experimental evidence that suggests this is a reasonable model, and we have provided evidence to show indeed R1 alpha phase separation is critical for cyclic MP compartmentation. But I do want to share with you a little bit more data on demonstrating the sort of disease relevance of this model. And here we're looking at a fibrolamellar carcinoma. This is a rare liver cancer that mainly affects young adults. So Sandy Simon's lab at Rockefeller a few years ago did a pioneering work to show the oncogenic fusion protein between R1 alpha, between DNAJV1 and PKCAT is present in almost all FLC patients. This suggests that this is the culprit. This is the genetic alteration that contributes to the tumorigenesis. However, the molecular mechanism were not well understood. We show this fusion protein potently inhibit R1 alpha phase separation. As you can see from here in these model example images, if we look at these FP tagged fusion protein and FP tagged R1 alpha, in these two cells, there's quite a bit of fusion protein present. Then there's very little punctal formation. But in these two control cells, where there's no fusion protein present or very little fusion protein present, we have a lot of R1 alpha punctal formation. And shown here is the quantification. And indeed, the presence of this DNAJB1 CAT fusion potently inhibit R1 alpha phase separation. Without this R1 alpha phase separation, we do not have effective cyclic compartmentation. And we've shown loss of R1 alpha phase separation and cyclic compartmentation leads to enhanced cell proliferation and increase the cell transformation. Here, I'm only showing you cell transformation data. Basically, this is the soft ARGA assay where we look at anchorage independent colony formation as a cell transformation assay. You can see in wild-type cells, we get very little colony formation. And if we knock out R1 alpha, we got a lot of colony formation. And this suggests R1 alpha has a previously underappreciated role of tumor suppressing. But if we supply the wild-type R1 alpha back, we recover the wild-type behavior, no colony formation. But if we provide a R1 alpha molecule that either doesn't phase separates or phase separates a little bit, but does not bind to cyclic MP, we still get a lot of colony formation. So these data suggest indeed the loss of R1 alpha induces anchorage independent colony formation, which is an indication of increased cell transformation. So to summarize this part, we show R1 alpha phase separates at endogenous conditions and serves as a dynamic cyclic MP buffering system to enable cyclic MP compartmentation. We also show this oncogenic fusion protein in FLC potently inhibit R1 alpha phase separation, thereby disrupting cyclic MP compartmentation, which can lead to increased cell proliferation and cell transformation. Currently, we're looking at the broader implication of dysregulated cyclic MP PK signaling in different cancers, especially in pancreatic cancers. But in the remaining few minutes, I wanted to talk about another effort in the lab where we try to enhance the sensitivity of some of our biosensors to enable imaging in vivo. For some of the molecules that we study, it's very, it provides a lot of the information if we can go into a more physiological relevant systems such as in vivo system. For example, protein kinase A serves as integrator for a lot of the neuromodulatory activities. So we're interested in looking at a pKa activity in neurons in free, if possible, freely running mice. However, these experiments are very challenging because now we're dealing with optically more complex models or samples compared to living cells. And we're also looking at endogenous activities that are quite often just very subtle and show small changes. This suggests we need more powerful and sensitive fluorescent biosensors. So Soham, a project scientist in the lab, and Jin Fan, a graduate student in the lab, collaborated in generating single fluorophore-based pKa activity biosensors. Soham also demonstrated this approach works for a lot of different kinases. So the X-ray design works for potentially the whole CAR collection. So in this particular design, we're using single fluorophore, single FP as the reporting unit. Our sensing unit is still the same, which consists of a substrate for the kinase of interest and a phosphoamino acid binding domain. But now instead of coupling this to a Fred pair, we're coupling this to a circularly permutated grain fluorescent protein. For circularly permutated fluorescent protein, you simply take the FP DNA and swap the two N and C terminal fragments. At this 3D level, this still folds into a functional beta can, but now it's a little bit more sensitive to the immediate environment. So this is a way of sensitizing the reporting unit. So as you can see, we're coupling our sensing unit to this circularly permutated FP, similarly to the GCAM design, if you're familiar with some of the calcium indicators that are currently in the field. However, different from the GCAM, we got a excitation ratio metric sensor out of this design. Here, we're looking at the excitation ratio. We're basically taking the ratio of the two excitation peaks of the enhanced grain fluorescent protein at 480 and 405, and use that as a readout as a PK for pKa activity. And using this readout, we're able to get an 8-fold change in the signal when pKa phosphorylate this sensor. And we also show in cells how the control works. If we mutate the phosphorylation site or include a PK inhibitor, we can abolish this change. So this highly sensitive readout allows us to perform in vivo to photon imaging. This is in collaboration with Rick Hugener lab at Johns Hopkins. So we're looking at layer 2-3 neurons in the primary visual cortex in head-fixed mice. And this is experimental design, and this is the actual image taken from an experiment. So the mice were running on the treadmill, and we're looking at the forced locomotion and also follow the pupil area as the indication and for the forced locomotion. And then we're able to track pKa activity in these neurons in the primary visual cortex. And we saw highly correlated pKa activity, very much correlated with the forced locomotion. And as I mentioned, the pKa is an integrator that integrates a lot of the neuromodulatory activities. So we think this could be a general assay for looking at a lot of the neuromodulatory activities by using PK activity as a readout in vivo, just the way we're using calcium for electric activities in different living systems. And this allows us to connect the biochemical changes in vivo with the behavior changes of the living organism. So finally, again, to come back to our hypothesis, we think where we hypothesize, again, these cellular biochemical activities are spatially organized by organelles, or these membraneless organelles, and microdomains and nanodomains inside the cells. And we are interested in testing this hypothesis using some of the biosensor technologies and microscopy methods. We're also very interested in characterizing the dysregulated activity architectures in different diseases, especially in cancers. And shown here, finally, just to acknowledge people, I think I mentioned some of the names. I think I may have forgot to mention Brian Tanner and Soham are the main contributors. Together with Jason, developed the Fluorescent technology. I think I mentioned the majority of the collaborators, but also mentioned Pat Meany, a colleague at UC San Diego, is our expert in modeling. We're combining some of the quantitative data with computational modeling to provide a more quantitative understanding of cell signaling. And here are our funding agencies. And I'll stop here and see if there are any questions. Thank you.
Thank you very much, Jin. It was a great talk. And I also, I realize I missed a lot of stuff that you have done lately. So I will catch up. It's very exciting to see certain development. So that just while we give you some time to breathe and the people to post also further questions, we shift a moment to the Mentimeter, which I will run for the first time and therefore is likely to go very wrong. Do you see it? Do you see it?
Yep, got that.
Okay. Ooh, I lost the. Okay. We start very well. So for people can already log in Mentimeter. I don't see anyone at the moment. Let me check. So these are the things that usually go wrong, but
I feel your pain.
Do you have any suggestion about copy link is the right one?
Yeah, are you on full screen? Because I can still see your taskbar at the top there.
Okay, so what? Okay.
Yeah,
now sometimes someone joins.
Oh, yeah, someone's there, right?
So which is your? So anyway.
That's a great answer.
Full screen. Okay, I see it here.
Right. There you go.
Okay, so while people is logging in, let me say that this is the most famous, competitive, and uncertain quiz about microscopy and more. So so briefly to remind you that the rules are no answer or wrong as no point, no answer, no point. The faster you answer, the more points you get. As usual, the quiz master is always right, even when they're wrong, but they're anyway never wrong, and I'm the quiz master. So you will have a few minutes to join us, after which we will start the questions. We try to wait to join about kind of 10-ish people, if possible. Remember that the winner, we can get a foldoscope. And the idea originally, because everything started during one of the first lockdowns, was to provide the audience with foldoscope at home so that they can continue their work at home with this very super high-end microscopy tools. So anyway, I think we can go ahead if I know how to do. Okay, remember you need to answer very fast. And the first question is, for FRET-based biosensors discussed in the seminar, the FRET pair usually serves as the reporting unit, the sensing unit, the signal amplification unit, and there is only one answer that is the correct one. 4 seconds to the end of the voting. And the reporting unit, well, the split of 3 for 2. Remember, you need to answer as fast as you can. And in the leaderboard, there is Frosty, followed by Android. Oh, Android, oh, I see. So next question. The kinase activity reporter KAR is or contain a modified kinase, a surrogate substrate of the kinase of interest, or a transcriptional reporter that acts downstream of the kinase of interest. 3, 2, 1. Times is up. Times is up. And again, there is a good split of people. So it is a surrogate substrate of the kinase of interest. The leaderboard is going to change. Stinglicious Ashley are going up, and Carl Gustav is now the first on our leaderboard. I feel I can do this as a profession from now on. Anyway, question 3, the CAMP compartmentalization model was initially proposed close to 40 years ago, close to 30 years ago, or close to 20 years ago. Again, only one answer is the correct answer. 4, 3, yeah, 2, 1, 0, time's up.
Is it paused?
Oh, Ali, we've lost you. Oh dear, we've lost our quiz master, and there's nothing. Oh no, here we go. Are you back again, Ali?
I think you're muted.
Ali,
Hi.
you're muted. There you go.
Sorry, this has never happened before. It's nice to have something new. I'm not sharing anymore, I guess, yes?
Nope, you're completely out of the quiz now.
Is the quiz running?
I can't see anything at the moment.
Oh, it's a pity. I thought...
No, it's not running. It's paused at the answer.
No.
There we go.
Okay.
Yep, we're back.
Sorry for that.
It's
I
gone,
don't remember.
it's gone into
Okay.
the next question.
So, okay, question number 5. X-ray series of kinase activity reporters use the following as readout. FRET, ratios of the excitation peaks of a donor FP and an acceptor fluorescent proteins, or ratios of 2 excitation peaks of GFP. And the right answer was ratios of the 2 excitation peaks of GFP. So only one person got it right. So the final leaderboard is, Android probably was the only one remaining to answer, but fully deserved for her previous answer anyway. She's on the top of the leaderboard. And please make yourself known on the chat, and we will send you the foldscope. And again, apologies for the interruption of the communication. It was very unfortunate for once that I do the Mentimeter. Nick, please go with the question and answer.
Yep. Okay, so if you could actually unshare, but if we kick off with Miranda, are you actually in the audience still? So if you want to unmute and ask the question yourself, you are very welcome to do so.
Hello. Are you able to hear me? Okay.
Yep, we can hear you.
Thank you for the talk. It's really interesting. I was just wondering, you were saying it's kind of this pKa sensors similar in ways to GCAMP? Have you looked at using it as a measure of memory formation, like either in vitro with LTP methods or during train spacing when looking at long-term memory?
Yeah, indeed. This is the one, I guess, actually after publishing X-ray ECAR has been requested by a lot of the neuroscience labs as part of the effort. So pKa activity can be looked at together with some of the other biochemical activities during the, for example, LTP process, because pKa phosphorylate, for example, AMP mPAR receptor, and as a critical step for the memory formation.
Thank you.
Okay, we've got another question from Vijay Dravmiam. Are you there, Vijay? If you want to, hi.
Thank you, Nick. Hi, Alison. Hi, Jin. That was a fantastic talk and a lot of.
Thank you.
I have a question about the split GFP. So, you know, do you have pools of molecules that have not formed the complex? Because you have one which is like a shorter part of the protein, and then there is one that's very long. Do they find each other happily? How is it in cell? And once they find each other, do they stay together or can they split again?
Yeah. For the split FP, this particular pair was sort of engineered and optimized, so they do sort of reconstitute pretty effectively. And once they find each other and reconstitute into a functional superfolder GFP, they probably will stay together. You know, we're very interested in reversible split FP, but I don't think we have good evidence in the field that there's a reversible pair available yet. Okay. So yeah, it's probably gonna stay together. And this is very different from, maybe some people in the field are familiar with the BIFC approach early on that was developed for looking at protein-protein interaction. In that case, the two pairs need to be, or the two fragments need to be brought together by a interacting protein pair. But this is different. This particular two fragments will just find each other and do so pretty effectively, I would say.
Excellent. Thank you so much.
Sure, right. Thank you.
Sorry, can I just to follow up? Because I don't remember this GFP. I don't remember. It's called GFP 11 or Delta 11. You say that is irreversible. Is it reversible because of high affinity, or because also like for bimolecular complementation, the chromophore is formed after the two halves meet each other? So in other words, is a covalent binding or is it simply high affinity binding?
Yeah, I think at least the current evidence suggests, like these two have to find each other for the actual maturation of the fluorophore.
Okay.
So, and once it's formed, that's relatively irreversible.
Okay, thank you.
All right, so any more questions out there? Not so much. I've got a question, sort of much more general. So obviously there's a lot of sensors around, and you never really sort of hear about all the engineering that goes on in the background to actually make them work. So do you have a sort of rule of thumb for sort of a minimum delta F over F value for something that you'd actually want to go ahead and use?
Yeah, so we actually, for some of our first generation sensors, they have very, pretty poor delta F over, especially delta R over R, because some of our initial sensors are FRET based. For example, the one that we developed and used quite a bit already is a sensor for mTOR complex 1 activity. We try to enhance it using different approaches, and you improved a little bit, but right now it's still just 15-20% change. but with careful controls, we are able to use it. And actually, this is the sensor that provided a really good evidence for us to discover this nuclear pool of mTOR complex 1.
Right.
So we think, especially if it's a ratio metric, you know, in the range of the 15%, 20%, we can use it. For delta F, you know, if it's a intensity-based sensors, maybe it's a little, we need a little bit more, because
Right.
as you may know, right, like we don't cancel out, we cannot use the ratio to cancel out some of the variations or the, you know, changes of cell shapes and stuff like that. So for that, we need a higher delta F over F. But for the ratio, I think, yeah, like 10% is our starting point.
Right. Okay. Well, that's very, very useful to get a sort of opinion from someone who actually makes them, because you know, I mean, and plus also, you know, it's clear that some of them do evolve over time to improve.
Right. Yeah, that's definitely we love to, you know, we always try to improve it, enhance it. You know, hopefully there will be a second generation for this sensor as well to get to a more convenient dynamic range.
Great. Are there any more questions?
Actually, also I remember that in the conference where I met you, now is a few years ago in Belgium, someone was presenting calcium biosensors, and they did a you know, back of the envelope evaluation of the money that went into the development of calcium sensors, and which amounted to several millions dollars. And this is something usually neglected, that you know, there is a huge amount of effort, and very often all these development are done either in very specialist laboratories or from people that is doing as a side job, and therefore don't even have the, you know, the proper time to optimize. So either if something is working or it doesn't work and then is abandoned. So I think that it would be much better if there was, you know, if there were kind of funding specifically for sensor developments, considering that, you know, live biochemical detection in single cells is actually super important. Yes.
Right. Yeah. Definitely. Yeah. I echo that. So now we get a little bit better in terms of technology related funding mechanisms. Yeah. So compared to when I started my lab, it was really difficult to just go for the that aspect of the research. So yeah, it's getting better. I think Gigi has another question, right?
Yeah, go ahead, please.
Is it okay? Okay, I have just two tiny questions. I noticed most of your FRET pairs are, I could be incorrect because I might be just ignorant of your work. Mostly they are GFP, YFP, CFP, that kind of partners, not MK2, DS red, the M Sherry, the reds. The first question is, do you prefer these versus those for some reason? And the second question is, have you ever tried three-way FRET, in the sense protein X does something to protein Y and protein Y, that FRET becomes the, you know, the donor for another acceptor?
Yeah, thank you for your questions. So like for FRET pairs, even I guess nowadays in the lab, we still start with cyan yellow FRET pair. They're kind of a relatively well behaving, although, you know, may not be the most convenient to use for, you know, depending on, you know, what kind of filter sets you have for the scope. But we start with the cyan yellow, and we have quite a few different pairs that we typically try. Then we move to green red quite often. And for green red, we do use, you know, the different grains that the field now have, and the different reds. We test, you know, typically start with a cherry, although the re-emission of the cherry is relatively poor, right? But we still, we typically do have cherry and ruby and scarlet. We also last year published a stack RFP that we will also try. So quite a different green-red combinations. We don't use, actually we haven't used DS red or you know, TD tomato or you know, for some of those. For our typical sensors, we like to stay with the monomeric version. But you know, there are sensors that have been developed that have two acceptors, right? One donor, two acceptors. One of the CRUK sensors that have that design turned out to work pretty well. So it's a good possibility to try those as well. Like for example, TD tomato. And we just haven't used that design. And then for the three-way FRET, actually a few years now, probably quite a few years ago, we published a sensor that's kind of have two different sensing units and three different fluorescent protein. So like you know, FP1 sensing unit 1, FP2 sensing unit 2, and FP3. And it's kind of fun little sensor that allows us to look at the CRUK and pKa simultaneously as a sort of co-imaging approach. We have cyan, yellow, red. So cyan could, you're right, do FRET with yellow and also red, and yellow can do with red. So we do have to sort of do a little bit of a calibration and a control to figure out which is talking to which, but it's definitely doable, and it was a fun project.
Thank you, Jin. Thanks.
Sure.
Okay, well, I think we've got out of questions, so maybe I'll just hand back to Ali to close up today's talk. And
yeah, I mean, probably I use the last 1 minute just to ask. I believe, and I'm sorry if I'm wrong, that you don't use a technique like fluorescence lifetime image microscopy, and tell me I'm wrong.
we do use.
Okay.
We do use.
Okay. Sorry, I haven't noticed. I will have to research that. But I was wondering if now film will become more available because now there are more commercial systems and so on.
Yeah.
Do you have any interest in trying to, you know, optimize sensors specifically for film?
Yeah, definitely. We're very interested in considering that possibility. So as I mentioned, we do use film for some of our mechanistic studies, although I don't think we have published it. So
okay, this is why.
not your fault. But we and for some of the sensors, we collaborate with others to also, for example, we're wondering if the X-ray series, because the signal is really large, and we wonder if there's any lifetime change that can that be also used together with the lifetime measurement. But we haven't focused on enhancing the sensors based on the lifetime. I think there are labs that focus on that, and it's, as you mentioned, the lifetime-based measurement now is getting much, you know, widely used. So it's definitely a very it's a great approach. So we'd love to incorporate that into our sensor design.
There is some work. There is not there is not a huge amount of work, and for us, what often is disappointing is to see that sensors that can be working with the ratiometric FRET, which I personally use for a lot of the biological questions we have, but being, you know, a developer of film systems, is very disappointing when we see that those sensors do not work with film, and is actually most of us do not understand why, because they should. So I think there is a huge amount of scope for improvement and to, you know, harvest the benefits of those technologies eventually.
Right, definitely. For example, for our cyan yellow pair, if we need to go to a film-friendly cyan for the straightforward lifetime measurement rather the complex lifetime profile. Yeah, so we do have that in mind for some of our applications. But yeah, I agree. Definitely need to incorporate that considerations more in the sensor design.
Okay, so thank you all. Thank you who remained until the end of the questions. Thank you, Jin, for the early talk in US times, and I'll meet you all next week, hopefully. Thank you, Jin, again. We'll stay in touch.
Thank you again for the opportunity. Great.
Bye.
Everybody.
Bye.